Good evening, everybody. I'm assuming that my screen is clearly visible to everybody, and I am audible clearly. Okay. Well, I'm launching technical poll to check for the technical things whether I am audible clearly and my screen is clearly visible to you or not. Then we'll be starting the class. Okay. So if you can hear me properly and if my screen is visible to you, then click on yes option. From JNV Haveri. Good evening, everybody. Good evening. Good evening. Yeah. So if you can hear me properly and if my screen is visible to you, then click on yes option. Guys, I've launched technical poll. If you can hear me properly and if my screen is visible to you, 1 second. Okay. Hopefully you can see me properly. Sorry, you can see my screen properly and I am audible clearly. Good evening, Ashwini. Not audible clearly. Once again. I hope I must be audible clearly. Am I audible clearly? And is my screen visible to you? Okay. I'm ending the poll that, and I am assuming that I am clearly audible. Well, I feel like there is a network issue here. Anyway, I'm launching the poll again because people are saying that my voice is not clearly audible. So that's why I'm checking it 1 second. Is my voice clear now, Ashwini? Am I audible clearly? Am I audible clearly? Yes or no? Hello, Jeffrey. Good evening. Okay, I'm assuming that I'm clearly audible and my screen is clearly visible to everybody. Okay, now I'll be ending the poll and we'll start the class. Hello, Mohammad Raja. Great. Okay, then let us start the class. Okay, so whenever we are talking about the particular chapter, human health and disease, so in this chapter, we are done with most of the things. Only few things are left, that is a disease that is related to human immune system. उसके बारे में हम लोग बात कर रहे थे. ठीक है, वहीं से कंटिन्यू करेंगे और उसी को कंप्लीट करेंगे आज की क्लास में. ओके, see guys, I'm just taking a lot of time in this particular chapter because people, they ask me, like some of the students, they ask me to go slow in this chapter because they need this chapter once again. So that's why. Okay, well, so let us talk about this particular section. अभी के बारे में बात करें तो हमने क्या देखा है और क्या हमको देखना बाकी है. So let us start with a question. This particular question, the question is like, AIDS is caused by, okay, option A, blood cancer, option B, STLV3, option C, bacteria, option D, TMB. Okay, well, I'm launching poll for the question and I hope this is a very easy question for you people to answer the, right? So I'm assuming that this is a very easy question to answer. Well, so I have launched the poll for this particular question and 33% people have written their answer. Jayasri, very good. Jayasri, Jayawalan, you have attended my last class and because of that, I guess you can answer it properly. Okay, 66% people have chosen their answer. Last 40 seconds remaining. 20 seconds gone. Okay. Last 30 seconds remaining. Only 66% people have chosen their answer so far. I want rest of the people to answer to the question. Mohammad Raja, Jayasri, and Ashwini from JNP Haveri. Hello, good evening, everybody. Hello, hello. Okay. Well, so last 10 seconds remaining, then I'll be ending the poll. Answer to the question, guys. Do it first. Very less time is left. I'll be ending the poll now. Last 5 seconds remaining. Okay, I'm ending the poll now. Yeah. So I'm sharing the result with you people. Result is like nobody have chosen option A, C, and D. Option B is chosen by everyone. Let me stop sharing. Then I'll explain you this particular question. Okay. AIDS is caused by option A, blood cancer, option B, STLV3, option C, bacterium, option D, TMV, tobacco mosaic virus. Okay, when you are talking about this particular question, AIDS is caused by a virus that is HIV. और यहाँ पे तो HIV का नाम लिखा नहीं है कहीं पे भी HIV वायरस, ठीक है, तो फिर तो कंफ्यूज हो गए, ब्लड कैंसर के वजह से तो नहीं होता, ये तो हटा दो, ठीक है, बैक्टीरियम नहीं होता, ये भी हटा दो, TM भी नहीं होता, टोबैको मोजाइक वायरस भी नहीं होता, बचा क्या है अपना STLV3. What is the full form of STLV? Can anybody tell me in the chat box what is the full form of STLV? STLV का मतलब क्या है? Can you expand STLV? What do you mean by STLV? STLV. Can anybody answer me in the chat box? Human TLV. Human TLV I told. That is human T lymphocyte virus. T lymphocyte virus. Okay, so this is human T lymphocyte virus. This is human T lymphocyte virus. It is going to attack the T lymphocyte of the body and it causes AIDS disease. STLV3. Other than STLV3, many more category of HIV viruses are there. And like HCLV, one is your HCLV. Okay, it is human. Hello Lohit from JNB Averi. Okay, that is STLV, and one more is like HIV1 and HIV2. These are many different category of viruses. HIV1, HIV2. If you compare HIV1 and HIV2, one is the virus, HIV1 is the virus, okay, which is the most dangerous virus, and it is caused in India. It is mostly popular in India. When you are talking about HIV2, it is popular in Africa, and it is not that much virulent. इतना ज़्यादा virulent नहीं है HIV2. HIV1 जो है वो virulent है, बहुत ज़्यादा, मतलब I can say ताकतवर है, you can say, right? So I'm moving to the next slide, and in the next slide we will be talking about the transmission. Before that, let me go back to the previous slides. Structure of the HIV virus, जो कि मैंने बताया था previous class में. Talk about this particular structure of the HIV virus once again. इसको और एक बार बात कर लेते हैं इसके बारे में. When you are talking about the structure of the HIV virus, this is the structure of the HIV virus. The genetic material of the HIV virus are RNA, single stranded RNA. HIV वायरस रेट्रोवायरसेस होते हैं और इनका जेनेटिक मटेरियल जो होगा वो RNA होता है. The genetic material for this particular viruses are RNA. Okay, these viruses are RNA, not rRNA. It is RNA. Okay, so it is single stranded RNA. The genetic material of these viruses are single stranded RNA. From here, again the DNA are synthesized. Okay, and for the synthesis of the DNA from RNA, we need some enzyme. कुछ एंजाइम चाहिए. तो RNA से जब DNA बनेगा तो उसके लिए क्या चाहिए हमको? RNA से when it is going to prepare DNA, that DNA is called as cDNA because that is complementary DNA. cDNA बनेगा और ये बनेगा एक एंजाइम के मदद से, that is reverse transcriptase enzyme. एक एंजाइम चाहिए, that enzyme is your reverse transcriptase enzyme. One enzyme that is called as your reverse transcriptase enzyme. So enzymes required are reverse transcriptase, proteases, and integrase, which will keep all the parts of the particular HIV virus together. That is your integrase. So RNA will be there, integrase will be there. You can see here integrase is there. Okay. RNA genome is there and reverse transcriptase enzyme is there. This is reverse transcriptase enzyme. Okay. Rather than that, if you are talking to this particular genetic material, a coat will be there which will be covering, that is nucleocapsid, which is again made of protein coats. Okay, and that protein coat is made up of P24. Inner coat जो होगा वो कितना कैसे बना होगा? P24 से बना होगा this inner coat. And the outer coat, which is made up of lipid, is made up of again like P18 material. P18 material, protein 18, P18 material, P24 material, P18 material. ये जो होगा ये proteinaceous होगा. Hello Chetan. So this is proteinaceous होगा, and this will be the lipid material. Outer one will be the lipid material. Okay. Okay. Core is covered by lipoprotein GP120 and GP41. So complete lipid layer के चारों तरफ this particular structure is made up of GP120 and GP41. The upper head-like structure, this is made up of GP120, and the base-like structure, this particular base is made up of GP41. It is made up of GP41, right? So this GP41 and GP120 together forming a structure, and that structure is going to help getting attached to the macrophages. ये जो स्ट्रक्चर, ये दोनों मिलकर के एक स्ट्रक्चर बनाएंगे जो कि मैक्रोफेजेस के साथ अटैच होगा. मैक्रोफेजेस के साथ ये अटैच करने में हेल्प करेगा. Get attached to the macrophages. Is that clear to everybody? आया समझ में सबको? So this is all about the structure of the HIV virus. Genetic material, single stranded RNA, enzymes, reverse transcriptase, integrase, globulin protein, JSP, that is GP4 globulin protein, which is a globular cell, or I can say spherical cell, globular protein GP. Okay, that is made up of lipoproteins, as I told you, globular proteins. Okay, I am moving to the next part now. This particular GP120 has complementary sequence to the CD4 receptors on macrophages and helper T cells. So as I told you, what is the role of this particular GP120? GP120 get attached to the two things like CD4 cells agar rehta. So the role of the CD4 cell is to getting attached to the macrophages and helper T cells. But GP120 will be like will mimic the role of CD4. So GP120, instead of like CD4 is not there. So instead of CD4, this GP120 is going to get attached to the two things. First one is your macrophages and second one is your helper T cell. Macrophage and next one is your helper T cell. इसके साथ ही attached होगा. So that's all. This is all about your structure of the HIV virus. Okay, I'm moving to the next slide now. Before that, if you have any kind of doubt, just let me know. And if you don't have any doubt, click on yes option. Jayshree and Mohammed. Great. So take a screenshot. Now I'm moving to the next slide, and in the next slide, you can check what are the things that is there. देख लो, अगर चाहते हो तो मैं अलग से बना सकता हूँ, कोई दिक्कत नहीं है, लेकिन I don't think we need to make it once again. Reverse transcriptase, integrase, protease. I don't need to make that one. P18, P24, GP120 lipid layer and other things. Okay, so these are the protein codes. If you want me to make this one once again, I can show you how the things are made. It's very simple. In the inner part, this is the genetic material RNA, right? And just it is single stranded. Just near to this particular RNA, reverse transcriptase enzyme will be there. So I'm making that one, that is reverse transcriptase enzyme. I'm doing it a little bit faster. Reverse transcriptase. Okay, and this particular structure is your RNA. RNA. इसके पास प्रेजेंट होगा कोई दूसरा स्ट्रक्चर, मतलब रेड कलर का मैं बना देता हूँ, इधर एक एंजाइम प्रेजेंट है और ब्लैक कलर का एक एंजाइम ऐसे बना देता हूँ, जस्ट टू डिफरेंशिएट दिस पर्टिकुलर ब्लैक कलर एंजाइम. रेड कलर एंजाइम इज योर, लाइक इसको मार्क कर देते हैं. ओके, सो सपोज दिस इज योर प्रोटीएज, तो दूसरा वाला जो बचा है वो बनेगा और एक एंजाइम, दैट इज ओके, प्रोटीएज एक है तो दूसरा वाला और एक बनेगा इंटीग्रेस. सो ये कुछ एंजाइम्स है, इंटीग्रेस. 4 एंजाइम का नाम याद रखो. चैलेंजिंग जस्ट कवरिंग रहेगा, इन सब के चारों तरफ एक कवरिंग रहेगा, ऐसे एक कवरिंग रहेगा, प्रोटीन का कवरिंग. सो दिस इज P24 कवरिंग, और इनके चारों तरफ अनदर कवरिंग रहेगा, अनदर कवरिंग विल बी देयर ऑल अराउंड दिस, एंड दिस इज अगेन ए कवरिंग व्हिच इज मेड अप ऑफ लाइक P18, और दोनों के दोनों प्रोटीन कोट होते हैं. All these two are like protein coats. Above this protein coat, another layer will be there. I am making that one in black color. So एक और कोट होगा इन सब के ऊपर ऐसे, and that coat is again like इसको कहा जाएगा अपना lipid coat. And on the lipid coat, now what happens on this particular lipid coat? A head-like structure will be there like this, and a tail-like structure will be there, and a base-like structure will be there. Can you see this one, right? So this particular head-like structure is your GP120, which identify and attach to the, which identify and get attached to macrophages, identified and get attached to macrophages, and macrophage के साथ ये attached होगा, और किसके साथ? And to the T lymphocytes, T lymphocyte के साथ. इसके बेस में, इसके बेस में प्रेजेंट होगा GP41. This is different. So this is the whole structure about the structure of the I can say HIV virus. ठीक है, ये GP120 और GP41 का जो स्ट्रक्चर है ना, ये चारों तरफ प्रेजेंट होगा. MSC कॉम्प्लेक्स होगा, मल्टी हिस्टो कॉम्पेटिबिलिटी कॉम्प्लेक्स, वो अलग चीज होता है, वो अभी के लिए नहीं है, उसको हटा दो दिमाग से. So this is all about the structure of the HIV virus. HIV virus. Well, I'm moving to the next slide now. Okay, so ये तो मैंने बता दिया, ये क्वेश्चन हमने सॉल्व कर लिया है. Now we'll be talking about the mode of transmission of disease. ये डिजीज कैसे ट्रांसमिट होता है? How HIV get transmitted from one person to another person, not exactly one person to another person. We can say how it get transmitted from a unhealthy person to a healthy person. एक अनहेल्दी पर्सन से हेल्दी पर्सन तक ये कैसे ट्रांसफर करेगा, इसके बारे में हम लोग बात करेंगे. ओके, ये हटा देते, पूरा हटा देते इसको. ओके, सो नाउ यू कैन चेक दिस वन. ब्लड, लिम्फ एंड सेरम अगर है तो इसके अंदर HIV वायरस होता है, और इसी के थ्रू ये ट्रांसमिट हो सकता है. ब्लड से बॉडी फ्लूइड्स लाइक ब्लड, lymph and serum, in that HIV virus will be there, and through this HIV virus can be transmitted. If a contaminated blood is there, if a contaminated lymph is there, and if a contaminated serum is there in which HIV virus is residing, that can be contagious, like that can spread the disease. But if you are talking about saliva, urine and sweat, इसके अंदर बहुत लेस अमाउंट के होते हैं, HIV वायरस बहुत कम अमाउंट के होते हैं, which is not that much useful, like which is not I can say यूरिन में, स्वेट में, सलाइवा में इतना कम HIV वायरस होते हैं, which is not that much quantity की मतलब spread हो सके, not spreadable, we can say not spreadable, spread नहीं कर सकते, इतना कम होते हैं. Now if you are talking about mode of transmission, remember this one, three things: blood, lymph and serum can be the carrier, or where more amount of HIV virus will be residing. Okay, and through this blood, lymph and serum, contaminated blood, lymph and serum, HIV disease can be transmitted. Okay, there are 4 different routes for the mode of transmission of the HIV disease. Okay, if you are talking about sexual route, sexual intercourse, contact with infected person, or sexual contact with infected person can leads to the transmission of the disease. As you can see in the first particular diagram, ये picture में देखो, this is the sexual contact. Like when two persons are having sexual intercourse or sexual contact with one another, and one person is affected, or then there is a higher chance, or we cannot say like chance only, there is we will get security that the particular disease will be transmitted to the healthy person also. So sexual intercourse or contact with infected person leads to the transmission of the disease. This is the first route. Second route, parental route. What is that parental route? Through contaminated blood transfusion, tattooing infected, poorly sterilized dental instruments. So this is parenteral roots. Parenteral roots मतलब क्या है, ये थोड़ा सा different है. Whenever a particular instrument that is used by the patient will be used by another healthy person. So what will happen in that case? If anything that is used by the patient is again used by the healthy person, then there is a chance of the transmission of the HIV disease due to that contaminated instrument. Okay, the contaminated blood transfusion, I mean, like, this is a person I am telling, this is a person, this is another person. So sometimes what happens, due to accidental cases or due to any other reason, there are needs of the transfusion of the blood. So whenever you are talking about transfusion of the blood or blood is required to a patient, okay, so this one you can see blood transfusion. Blood is required for a patient. Before we should transfuse the blood to the patient, before we should transfer the blood into the patient, before we should inject the blood into the patient, we need to understand, check, and know the blood group of the patient as well as the donor. We need to check whether the donor is HIV positive or not, and after that only they are allowed to go for the transfusion of the blood. So blood transfusion किया जाता है, at that time ये चेक किया जाता है कि वो मतलब HIV positive है या नहीं. अगर है तो फिर allow नहीं किया जाएगा, because that is there is a chance of transfusion of the disease through that. Okay, because in the blood, lymph and serum there is a huge or high amount of HIV virus that accumulates. Okay, blood transfusion से, tattoo बनाने से, tattoo बनाने से. So tattoo बनाने से क्या होगा, जो इंस्ट्रूमेंट यूज किया गया होगा टैटू बनाने के लिए, उस इंस्ट्रूमेंट में इन्फेक्टेड पर्सन का ब्लड लगा होगा, और उसी को लेकर अगर फिर से किसी टैटू बनाते हैं हेल्दी पर्सन का, तो वही ब्लड से होकर के पास कर जाएगा, कंटामिनेट हो गया होगा वो इंस्ट्रूमेंट, एंड थ्रू दैट कंटामिनेटेड इंस्ट्रूमेंट नाउ दिस disease can be transferred. Okay, or disease can pass. So that is tattooing infected, poorly sterilized dental instrument tattooing से होगा, and infected, poorly sterilized dental instrument, I mean, like, dental instruments will be there, and this dental instrument needs to be cleaned properly before and after its use. Dental instruments are very costly, and the dentist who generally use this particular dental instrument, they need to sterilize it, clean it properly before and after use of the. My voice is lagging. Okay, Mohammed, 1 second, let me check. Is happening? Jayasri, is my voice lagging? Jayasri, let me know in the chat box. Is my voice lagging? मेरा voice lag कर रहा है क्या? Is it clear now, Mohammed? Jayasri, still lagging. Yes, little lagging. Okay. Got it. Let me check the connection there. अभी मेरा वॉइस क्लियर होगा, राइट? आई गेस अभी मेरा वॉइस क्लियर होगा. जस्ट लेट मी नो because there is an internet issue. So is my voice lagging now also, or it's clear? Mohammed, Jayasri and Srusti. अभी मेरा वॉइस क्लियर है और it is still lagging? Let me know in the chat box whether clear. Okay, then we'll continue from here. So tattooing करने से होगा dental instrument, like dentists they usually use instruments. Okay, and if a person they are using it against a person who is infected with HIV. Okay, now without cleaning it properly, if that particular instrument is used for another patient who is healthy, like healthy against the HIV virus, so there is a chance of the transfusion of the disease through this particular unsterilized dental instrument also. So that's why dental instruments needs to be cleaned before and after use by the dentist. Okay, Mohammed, thank you. Right. So next, third one is your transplacental route. Transplacental route from mother to fetus through placenta, from mother to infant through colostrum. So transplacental route means from mother to the baby or to the I can say fetus. The disease can transfer through two different way. First one is through placenta. From mother to fetus, there is a chance of transfer of the disease through placenta. Okay, and from mother to the infant through colostrum, and colostrum is present in the milk. Colostrum is present in the milk. Okay, so disease can also spread through this. Now, what happened? The voice is buffering. One second. I hope the voice is clear now. Is that so? If the voice is clear, then please vote. I'm launching teacher NPS poll once again. Just let me know through the teacher NPS poll if my voice is clearly audible. Only clearly audible. If it's not lagging. Great. अभी clearly audible होगा मेरे ख्याल से. ठीक है. So my voice is clearly audible now and I can continue the class. If it's happening once again, just let me know during the class hour. Okay. Within this class itself. Well, so through transplacental route means the disease can transfer from mother to the either fetus or to the infant. So how to the fetus through placenta, that is already mentioned here. Okay, and to the infant through colostrum. Colostrum is present in the milk. Colostrum, the color of the colostrum is yellow, right? Why this particular colostrum is very much necessary, Muhammad and Jayasri? Just let me know in the chat box why this particular colostrum is very much necessary for an infant. For an infant, किसी भी infant के लिए, why this particular rich in antibiotics? Exactly, Jayasri. It is rich in antibiotic. Exactly. Okay. So, well, if it is rich in antibiotic, which particular antibiotic? Just let me know. It is rich in antibiotic, I got it, but which particular antibiotic? It is rich in which antibiotic? Great. Are you sure it is reached in IgM, Mohammad Raja? Two things remember. अभी फिर से मैं बता रहा हूँ वो दोनों चीज को. एक याद रखो, जीपी ग्राम पंचायत, IgG will be transferring through placenta, and another is your AM. Okay, that IgA will be transferring through mother's milk. Okay, IgG through P, like P for placenta, IgA through milk, M for milk. Okay, GPAM इतना याद रखो. ठीक है, सो मिल्क के थ्रू ट्रांसफर होता है IgA, not M. ठीक है, through infected needles. If a person is infected and a needle is used for that particular person, और उसी needle को लेकर के दूसरे person में इंजेक्ट करोगे, so there is a high chance of mode of there is a high chance of transmission of this particular disease. Okay, IgA exactly. Well, I am moving to the next slide. चलो इस पर्टिकुलर क्वेश्चन इसको देख लो एक बार, इसका स्क्रीनशॉट ले लो, फिर हम लोग दूसरे स्लाइड में चलेंगे. टेक ए स्क्रीनशॉट ऑफ दिस वन, note the things, take a screenshot of this one, and just let me know if I have to move to the next slide. Then click on yes option, or if you are having any doubt, just let me know. You should not have any misconception that how the disease is spreading. Okay, it will be spreading through sexual route, through parenteral routes, through placental routes, or through infected needles. Okay, it is not spreading through food or shaking hand or while kissing, because saliva में होता है, HIV virus होता है saliva में, but not that much. Remember this one. In saliva, HIV virus will be there. In sweat also, HIV virus will be there. In urine also, HIV virus will be there. But that number of HIV virus in the saliva, sweat, and urine is not that much. And remember this one, it's spread through blood, right? But it cannot spread through insect bite. Although an insect is sucking your blood and it is going to suck another blood in another individual, but उसके बावजूद भी नहीं होता है, क्योंकि there are certain enzymes or something वो सक करने से ब्लड के थ्रू वो HIV वायरस पास नहीं करते इंसेक्ट में, और वो जाता ही नहीं है. सो दैट कैन नॉट बी द रीजन. ओके, और पूल में नहाने से किसी के साथ ये स्प्रेड नहीं होता है. एक साथ बैठ के खाने से, while having food, meal, shaking hand, kissing a person, or through insect bite, or while bathing also, this particular disease is not spreading. It can reproduce by transferring by saliva. No, what is that? It can reproduce by transferring by saliva. I did not get it, Mohammad Raja. HIV cannot reproduce by transferring through saliva. Remember this one. Okay, now here is a question for you people. Okay, the question is asking about, I'm just removing this particular thing. The question is asking about only of the following four ways through which ads can be spread or ads can create. इनमें से, इन चार में से, sorry, read the question properly and then try to answer to the question. I'm launching poll for the question now. Read the question once again. Only of the following four ways through which ads can spread. Option A, infected needles and syringes. Option B, through mosquito bites. Option C, looking after AIDS patients, shaking hands and coughing. Well, I'm launching poll for the question and I hope you can answer to the question in the poll. Correct, Jayasri. Correct, Muhammad Raza. Great. 10 seconds gone and I got answer from all the people, those who are present in the Zoom, but we are waiting for the people who are in YouTube to answer to the question. Last 40 seconds remaining. I'll be giving you complete 1 minute to answer to the question. Guys, who are there in YouTube, try to answer to the question. Let me check what is the status of the people in YouTube. Yes, Manjunatha. Yeah, Apurva, exactly. Very good, Apurva and Manjunatha. Okay. So I'm going to the correct Apurva. Exactly, Apurva. Well, I'm ending the poll now, sharing the result with the people. And the result is like everyone have chosen option A. Option B, C, and D is not chosen by anyone. Let me stop sharing. Then I'll explain it to you. Sorry. Then I'll explain it to you. Okay. So now if you are talking about this particular question on only of the following four way through which ads can spread. So infected needed and syringe से spread हो सकता है, ये तो सही है. Through mosquito bites नहीं होगा, insect से नहीं होता है. Looking after AIDS patient को देखते ही spread नहीं होगा, ये भी incorrect है. Shaking hands and coughing इससे भी नहीं होगा. Shaking hands ये बचना है. Shaking hands से spread होता है कोविड. कोविड स्प्रेड होता है सेकिंग हैंड से, तो इससे बचना है. सो ऑप्शन ए विल बी द करेक्ट आंसर टू द पर्टिकुलर क्वेश्चन. ये सही होगा इस क्वेश्चन का आंसर. ऑप्शन ए, एड्स कैन बी स्प्रेडेड और एड्स कैन स्प्रेड थ्रू infected needles and syringes. पैथोजेनिसिटी एंड इन्फेक्शन, ये किसको अटैक करता है और किस तरीके से अटैक करता है उसके बारे में दिया गया है एचआईवी एड्स का. इट अटैक्स ऑन द मैक्रोफेजेस एंड द टी हेल्पर सेल or T cell or helper T cell, because these macrophages and helper T cells, they are the most important part of our immune system. अपने immune system का ये सबसे ज्यादा important part होते हैं ये macrophages and helper T cell. ये macrophages and helper T cell होते हैं ये सबसे important part होते हैं. Remember this one. Okay. क्योंकि यही जा करके क्या करते हैं हेल्पर टी सेल एंड मैक्रोफेजेस, दे आर द मोस्ट वन सेकंड. हेल्पर टी सेल एंड मैक्रोफेजेस ये सबसे इंपॉर्टेंट होते हैं. ठीक है. सो रिमेंबर दिस वन, मैक्रोफेजेस एंड हेल्पर टी सेल जो होते हैं ये सबसे इंपॉर्टेंट पार्ट होते हैं अपने इम्यून सिस्टम का, क्योंकि मैक्रोफेजेस आर गोइंग टू किल द पर्टिकुलर पैथोजन एंड हेल्पर टी सेल इज गोइंग टू इन्फॉर्म द बी लिम्फोसाइट अगेंस्ट दिस पर्टिकुलर इन्फेक्शन. ठीक है. वायरसेस दे एंटर इनटू मैक्रोफेजेस एंड रिलीज वायरल जीनोम. वायरसेस क्या करते हैं मैक्रोफेजेस के अंदर, पहले इन्फेक्शन कहां से शुरू होगा मैक्रोफेजेस से. सो वायरस दे विल डायरेक्टली अटैक द मैक्रोफेजेस एंड टी लिम्फोसाइट्स. दे एंटर इनटू द टी लिम्फोसाइट एंड मैक्रोफेजेस. दे रिलीज देयर जीनोम in that particular cell macrophages. So viral genome, it will be single stranded RNA. Single stranded RNA होगा. वही single stranded RNA get incorporated with the host genome. Host genome in the sense अपने body का जो genetic material होगा in macrophages. With that particular genetic material, it will get incorporated and it start producing its copies. अपना copy ये produce करने लगेगा. It start replicating or it start producing its copy. Okay, with the help of the enzyme that is reverse transcriptase. कौन सा enzyme की मदद से? It is reverse transcriptase. Okay, right. This result information as like this is single stranded RNA and it will be like replicating continuously, and this result in the formation of the a vast number of HIV viruses. It results in the formation of the vast number of HIV viruses from macrophages, and इसी की वजह से इसको HIV factory कहा जाता है. Remember this one, because the viruses are forming in the macrophages. Viruses are formed in the macrophages. That's why it is called as HIV factory. Remember, this is a very important question. Which particular cell or which particular part of the human body or host body is called as HIV factory? That is your macrophages. They are called as or regarded as HIV factory because in that macrophages viruses are produced. This result in progressive decrease of T lymphocytes. So इससे क्या हो जाएगा, धीरे धीरे इन्फेक्शन बढ़ता जाएगा और इन्फेक्शन बढ़ने की वजह से T lymphocytes का नंबर इंक्रीज करेगा. एंड एज T lymphocyte का नंबर जब इंक्रीज किया और डिक्रीज करेगा और डिक्रीज करने की वजह से दिस लीड्स टू वीकनिंग ऑफ द T lymphocyte. ये डिक्रीज हुआ और डिक्रीज करने से ये किसको लीड कर गया, वीक. weakening of T lymphocytes. T lymphocyte का weakening को ये lead करता है. Is that clear to everybody? I hope this is clear to everyone. Now I am moving to the next slide. Right? I am moving to the next slide now. If this is clear to everybody, click on yes option so that I can move to the next slide. Great. I am moving to the next slide now. This is all about your pathogenicity and infection of the HIV virus. यहाँ से question, एक question must question है, that is HIV factory in the host body, that is macrophages, inside which this particular thing will be produced. That is, that particular procedure is given here only. वो बता दिया गया है यहाँ पे, अगर देखोगे तो फिर समझ आएगा. देखो राइट, ये इन्फेक्शन किया CD4 सेल होता है. CD4 सेल के जगह पे GP120 होते हैं जो कि इसको मिमिक करते हैं और इसके साथ ये बॉन्ड बना के ऐसे कांटेक्ट में आ जाते हैं. नाउ दिस पर्टिकुलर वायरस विल बी लाइक सेल के अंदर घुसने के बाद आरएनए को छोड़ दिया. आरएनए को छोड़ दिया, रिवर्स ट्रांसक्रिप्टेज एंजाइम की मदद से दिस पर्टिकुलर आरएनए विल बी सिंथेसाइजिंग डीएनए, that is cDNA. That is why it is written here complementary DNA. C for complementary. Why it is complementary? Because it is formed from the RNA and it is not the exact DNA strand which we wanted. It is complementary to the DNA strands because RNA are formed from the complementary strands of the DNA. ठीक है, ट्रांसक्रिप्शन ट्रांसलेशन पढ़ा है तो पता चलेगा तुमको कॉम्प्लीमेंट्री स्ट्रैंड क्या होता है. ठीक है, सो फ्रॉम cDNA अगेन कॉपी ऑफ दिस पर्टिकुलर बनेगा ये. और ये इंटीग्रेज एंजाइम जो होगा, द रोल ऑफ दिस इंटीग्रेज एंजाइम, देखो हर एक एंजाइम का काम है. रिवर्स ट्रांसक्रिप्टेज एंजाइम जो प्रेजेंट था बॉडी के अंदर, किसके, वायरस के बॉडी के अंदर, दैट रिवर्स ट्रांसक्रिप्टेज हेल्प्स इन ट्रांसक्रिप्शन ऑफ द RNA into DNA. Now the DNA is formed. This particular DNA will go and bind to the host DNA. ये जो अपना ब्लू कलर वाला है ना, ये host DNA है. host DNA के साथ bind करेगा. At that time an enzyme called integrase, if you remember, an enzyme called integrase which was present in the HIV virus. Thus the role of this particular integrase enzyme is to make a connection between host genome and viral genome. So viral genome and host genome को ये connect कर देगा. Remember this one. Integrase का काम क्या है, viral genome and host genome को ये connect कर देगा. So now you can see this is the viral genome, black color one, and the blue color is your host genome. Replication will continue and such in like after successful replication, many replication, again DNA will be produced. A lots of DNA, number of DNA will be produced here, and from this DNA RNA will be synthesized again, this black color strands, and from the mRNA, right, ribosome will be produced, P24, and from that ribosome protein will be synthesized which will forming the protein code, that is P24P18. इधर अपना RNA प्रेजेंट है, ये ब्लैक कलर वाला, और इधर प्रोटीन बन रहा है. सो ये सब मिल करके फिर प्रोटीन कोड बना के सबका असेंबली होगा. एंड आफ्टर ए कंप्लीट सक्सेसफुल असेंबली, इधर देखो सबका असेंबली हुआ, इधर देखो ये कंप्लीट लिखा हुआ है. ऑल दीज थिंग्स आर हैपनिंग इनसाइड दिस सेल. और इधर फिर सबका असेंबली हुआ, सब असेंबल हुए, और असेंबल होने के बाद व्हिच वन, दिस आरएनए व्हिच इज प्रोड्यूस्ड हियर, एंड सम ऑफ द mRNA ट्रांसपोर्ट इनटू द they went into the ribosome and they help in synthesis of the protein, that is P24 and P18, which will be forming the coats all around the viral structure. Coats को formation करने में help करेंगे. Yes तो no, all these things which are produced inside the human cell will be like they will get assembled at one point and they will be forming the more and more number of viruses. अभी बहुत सारे viruses को ये ऐसे बना देंगे. देखो एक वायरस सेल से कितने वायरस सेल्स बने, and all these things are produced inside the particular macrophage cell. and इसीलिए macrophage को क्या कहा जाता है, ये macrophage cells है. ह्यूमन सेल, कौन सा ह्यूमन सेल, macrophage सेल है, और इसी की वजह से इसको फैक्ट्री ऑफ HIV कहा जाता है. फैक्ट्री, HIV फैक्ट्री इसीलिए कहा जाता है इसको. ठीक है, आई होप ये क्लियर हो गया सबको. इधर भी लिखा हुआ है मैक्रोफेजेस HIV फैक्ट्री, राइट. I hope this is clear to everyone. Click on yes option now so that I can move to the next slide. The role of GP120 is to getting like attach करने के लिए किसके साथ, attach to host. होस्ट के साथ अटैच होने के लिए। नाउ आई एम मूविंग टू द नेक्स्ट स्लाइड as it is clear to everybody. That is whatever I told you there, that is only given here. देखो after infection what is happening, reverse transcription की वजह से ये ट्रांसक्रिप्शन हुआ, इंटीग्रेशन, और इंटीग्रेशन will be happening by the help of integrase enzyme, that is also given here. ये देखो ये जीनोम था, ये किसका जीनोम, डबल स्टैंडर्ड डीएनए डीएसडीएनए ऑफ द एचआईवी वायरस का डीएनए है, ये एचआईवी वायरस का डीएनए है, ये एचआईवी डीएनए है, दैट इज गिवन हियर, एचआईवी DNA है. This HIV DNA is going and it get attached or integrated to the insert into the host genome. ये host genome के साथ ये क्या हो गया, integrate हो गया by the help of enzyme integrase. Now replication will continue and HIV RNA will be produced at one side and in the other way ribosome से होकर के protease synthesize हुआ और they are going to make the protein coat and in this way new virus will be produced. This is the life cycle of the HIV virus. HIV virus का life cycle है ये, right? So that's all. This is all about your replication of HIV virus, how it's infecting and how its number is getting increased. इसका number कैसे increase हो रहा है? Remember this one question अगर आएगा HIV से तो question क्या आ सकता है? GP 120 का role क्या है? It will be doing similar role of like CD4. It will identify the macrophages and get attached to the macrophages. Second role, what is the role of integrase? Role of integrase is to integrate viral and host genome. Integrase का role क्या है? ये question आएगा. Integrase का role क्या है? To integrate viral and host genome. दोनों genome को integrate करने में ये help करता है. Third question, that is which particular organ is called as your HIV factory? HIV फैक्टोरी कौन सा है? इस सेल को HIV फैक्टोरी कहा जाता है, ये हो सकता है और एक आंसर, ठीक है, और एक क्वेश्चन ये बन सकता है, ठीक है. सो और एक क्वेश्चन बन सकता है कि HIV वायरस जो है, ठीक है, इसका जेनेटिक मटेरियल क्या है? व्हाट इज द जेनेटिक मटेरियल ऑफ HIV वायरस? ठीक है, दैट इज योर RNA, सिंगल स्टैंडर्ड RNA. रिमेंबर दिस वन, सिंगल स्टैंडर्ड RNA. ये क्वेश्चन है, ये क्वेश्चन, ठीक है. सो रिमेंबर ऑल दीज थिंग्स, दिस कैन बी आस्क्ड एज अ क्वेश्चन, ठीक है. 3-4 क्वेश्चन बन सकता है, देख लेना एक बार, ठीक है. अलॉन्ग विद दैट प्रोटीन कोड, अंदर वाला P24, उससे बाहर वाला P18, आउटसाइड दैट योर लिपिड लेयर. नाउ आई एम मूविंग टू द नेक्स्ट स्लाइड. दीज आर सर्टेन सिम्टम्स एंड consequences. In general, the HIV are asymptomatic. ये asymptomatics होते हैं, ठीक है. In general, HIV जो होते हैं वो asymptomatics होते हैं. Remember this one. Asymptomatics in the initial stage. Asymptomatics in the initial stage. Asymptom का मतलब क्या होता है? Symptoms दिखेगा ही नहीं. No symptoms. कोई symptoms ही नहीं दिखेगा, तुमको पता ही नहीं चलेगा कि तुमको disease हो गया है, ठीक है. No symptoms appear in this phase. This is asymptomatic phase. No symptoms appear in this phase. So let me take the marker. No symptoms appear in this particular phase. Okay. From few months to many year, almost like 2 year तक भी होता है. Few months to लेकर के 2 year तक कोई symptoms देखने को नहीं मिलेगा. Why? Because this HIV are detected by ELISA test. एंटीजन एंटीबॉडी टेस्ट करा के उनको डिटेक्ट किया जाता है और 2 से 12 वीक तक कोई भी एंटीबॉडी प्रोड्यूस नहीं होगा. नो एंटीबॉडी विल बी प्रोड्यूस्ड फ्रॉम 2 to 12 वीक. 2 से लेकर के 12 वीक तक कोई भी एंटीबॉडी नहीं है. तो इन मंथ अगर काउंट करें तो 12 वीक इन द सेंस 3 और 4 हिसाब से काउंट करें तो ऑलमोस्ट लाइक 3 मंथ तक, ऑलमोस्ट अप टू 3 मंथ there will be no sign of any antibody against the pathogen. Although the pathogen has entered into the body, but it will be like making itself that much adaptable so that our immunity could not identify the antigen. उस particular pathogen इतना अपने आप को अपने जैसा बना लेगा, अडैप्ट कर लेगा कि our own body, own immune system would not be able to identify the pathogen. And that is why there will be no production of any antibody. No production of any antibody cannot be detected through ELISA test during window phase. इसी टाइम पे ELISA टेस्ट करके हम लोग डिटेक्ट नहीं कर सकते because there is no production of the antibody. Antibody प्रोड्यूस नहीं हो रहा है ना. सो एंटीबॉडी प्रोड्यूस नहीं हो रहा है, इसी की वजह से एलाइजा टेस्ट के लिए हमको क्या चाहिए, एंटीबॉडी चाहिए, बट एंटीबॉडीज आर नॉट प्रोड्यूस्ड, दैट्स व्हाई इट इज कॉल्ड एज विंडो फेज। ओके, सो एंड दिस विल कंटिन्यू, ये कंटिन्यूअसली चलता रहेगा, तुमको पता नहीं होगा कि तुमको एचआईवी हो गया है करके। ओके, एंड दिस लीड्स टू ए वेरी डेंजरस कंडीशन, दैट इज देयर विल बी डिक्रीज इन टी लिम्फोसाइट कंसेंट्रेशन। सो टी लिम्फोसाइट धीरे धीरे घटता जाएगा, एंड दैट लीड्स टू एक्टिवनेस ऑफ द वायरस। इधर अपना इम्यूनिटी डाउन होगा, उधर अपना वायरस अपना जो रोल है ना वो दिखाता जाएगा। दिस इज इनिशियली इम्यूनिटी जो था वैसा स्ट्रॉन्ग था, वायरस एंटर किया, कोई दिक्कत नहीं, नाउ द वायरस इज रेप्लिकेटिंग एंड द वायरस नंबर ऑफ वायरसेस आर इंक्रीजिंग एंड आवर इम्यूनिटी इज गेटिंग डाउन बिकॉज ऑफ द लेस नंबर ऑफ टी लिम्फोसाइट्स। This leads to next phase, and that next phase is something different. So like इसको नोट बना लो और स्क्रीनशॉट ले लो, फिर आगे बढ़ता हूँ। First phase is your asymptomatic phase. First phase is your asymptomatic phase, right? I'm going to the next phase. Yeah, next is your AIDS related complexes, that is ARC. Okay, and then will be the final full blown AIDS. Okay, when you are talking about AIDS related complexes, it means there will be complexities due to the HIV characterized by certain symptoms. Swollen lymph nodes, the lymph nodes will get swelled off. Why? Because in the, this is the lymph node is the area, lymph node is the location, lymph node जो है ना ये swell कर जाएगा, I understand, I hope you also understand this one. Okay, why? Because in the lymph node antibody and antigen interaction happens for the first time, so इसीलिए ये swell करने लगेगा. Fever will be continuous. Okay, repeated episodes of diarrhea, कभी कभी डायरिया हो जाएगा. Repeated episodes of diarrhea मतलब अभी हुआ है तुमको, कुछ दिन ठीक हो जाएगा, again it will be like that only. Continuous weight loss होगा. Prolonged cough मतलब cough जो रहेगा वो तुम्हारा continue रहेगा, cough, prolonged cough रहेगा. I mean like prolonged means the cough will be continuously staying for years, for months. Okay, so these are certain symptoms which can be characterized as or considered under ARC, AIDS related complexities or complexes. Okay, T lymphocytes counts become very low from 900 to ये decrease होकर के कितना हो जाएगा, 200 हो जाएगा, very less हो जाएगा. ठीक है, per liter blood के अंदर 10 to the power 6 लगा दो बस, that's it. Remember this one, 900 to 200 T lymphocytes का काउंट जो है इनिशियली नॉर्मल कंडीशन में 900 होता है. ओके, इट विल रिड्यूस्ड एंड इट बिकम 200 पर into 10 to the power 6, I mean like that is 900 million and it will reduce up to 200 million, that's it, per liter of blood. Okay, next this leads to the last stage, that is your full blown AIDS. This leads to a condition where our immunity completely get down, अपना immunity जो होगा वो low हो जाएगा, and that results in opportunistic infection. What happened now? The infection, like a lot of infection will get an opportunity to infect our body now. Why? Because why it is happening? Because one second. Okay, so why it is happening? Because our immunity is getting lowered, so there is a chance of for other antigens or pathogens to attack our body. So अभी अपना body weak हो गया, तो बाकी बाहर जितने सारे pathogen घूम रहे हैं, they have a chance to infect our body, and that particular infection is called as your opportunistic infection. Okay, dysentery, cancer of the skin, source of the herpes. Okay, then pneumonia, candidiasis of mouth, tuberculosis. These are like other disease which can be caused due to HIV virus. So remember this one, HIV is a virus which leads to AIDS condition, and AIDS is a condition that have all other diseases. मतलब इसमें जो इंसान होता है HIV से मरता नहीं है. Why it is dying? Why the particular person is dying? Because the person is getting affected by other pathogens. उसको दूसरा डिजीज होगा, Why? Because of the less standard of the immunity, because जो immunity है ना वो अभी क्या हो रहा है, डाउन हो रहा है उसी की वजह से. So I hope this is clear to everybody, and if it is clear to you, click on yes option so that I can move to the next slide. Great, I'm moving to the next slide now. So first one, asymptomatic phase, 2 से लेकर के ऑलमोस्ट लाइक फ्यू मंथ से लेकर के कुछ ईयर तक, 2 to 10 years तक ये नहीं मतलब दिखेगा कुछ भी. ओके, कोई भी सिम्टम्स तुमको देखने को नहीं मिलेगा. ओके, एंटीजन एंटीबॉडी एलाइजा टेस्ट करो विंडो फेज में तो पता नहीं चलेगा तुमको. दैट इज द विंडो फेज, इनिशियल फेज. देन द आर्क फेज विल बी कमिंग, दैट इज एड्स रिलेटेड कॉम्प्लेक्सेस. ओके, यहाँ पे तुमको कुछ सिम्टम्स देखने को मिलेगा, वेट लॉस, रिपीटेड एपिसोड्स ऑफ डायरिया, प्रोलॉन्ग कॉफ. ओके, ये सब देखने को मिलेगा. एंड लेटर ऑन दिस लीड्स टू लाइक less count in the lymphocytes. T lymphocytes घटता जाएगा, from 900 million to 200 million तक ये घट जाएगा. This leads to low immunity and opportunistic infection that leads to many other infections like dysentery, cancer of the skin, source of the herpes, pneumonia, candidiasis of the mouth, and tuberculosis. Okay, I'm moving on to the next slide. Next is your diagnosis. Okay, diagnosis में screening test होगा and confirmatory test होगा. ELISA test is the test which is there for the screening. This is a screening test. ELISA full form enzyme linked immunosorbent assay. Through this particular test we cannot simply like confirm that the person is affected with HIV AIDS or not, but confirmatory test is there, that is your Western blotting test. This is another one, Western blotting, to identify the protein. कौन सा प्रोटीन? एंटीबॉडी वाला जो प्रोटीन है उसको आइडेंटिफाई किया जाएगा कंप्लीटली. ठीक है, सो एलाइजा टेस्ट के बारे में बात करेंगे इन द नेक्स्ट क्लास, अलोंग विद दैट कन्फर्मेटरी टेस्ट, दैट इज योर वेस्टर्न ब्लॉटिंग के बारे में बात करेंगे. ओके, तो रिमेंबर टू थिंग्स, एलाइजा टेस्ट इज द स्क्रीनिंग टेस्ट एंड वेस्टर्न ब्लॉटिंग टेस्ट इज द कन्फर्मेटरी टेस्ट. ये दोनों एक एक सवाल है. दीज आर द क्वेश्चन इटसेल्फ, क्वेश्चन इन देम. ओके, सो रिमेंबर दिस वन, two way of diagnosis, screening test, confirmatory test. For screening, ELISA can be utilized, and for confirmatory test, Western blotting can be done. Okay, right? इसके बारे में डिटेल में देखेंगे हम लोग. ट्रीटमेंट की अगर बात करें तो ट्रीटमेंट is to prolong the life of infected person. I mean treatment, there is no proper treatment for this particular disease. ट्रीटमेंट नहीं होता है, लेकिन we can increase the life duration, or we can say that is your lifespan of an individual infected person by providing medicines. Reverse transcriptase inhibitor, that is your zidovudine, stevivudine, DDI. Okay, didexamidine, protease inhibitor, that is your ritonavir, nelfivir, sequinavir. Okay, highly activated antiretroviral therapy include both inhibitor for reverse transcriptase and protease enzyme drug. So HART दिया जाता है, protease inhibitor दिया जाता है, reverse transcriptase inhibitor दिया जाता है. Okay, highly activated antiretroviral therapy, that is HART, highly activated antiretroviral therapy. These are certain medicines which can be given. Okay, well, so that's all for today. We'll be talking about the other cancer disease tomorrow. ठीक है, ये क्वेश्चन भी कल ही सॉल्व करेंगे. Okay then, so if you have any kind of doubt, just let me know in the chat box. I'll explain it to you. If you don't have any doubt, then please vote for me in the teacher LPS board. Yes, if you have any doubt, just let me know in the chat box. Okay, thank you. Thank you. Happy week.